And welcome to the IB Biology IA analysis video. So, what should be included? Your quantitative raw data, your quantitative process data, qualitative data, you need to do a stats test, and there should be a graph. We're going to go through each of these sections in detail. So, your raw data should all be displayed in a single data table, so not lots of different tables, just one single data table. The units should be in your column headings, and there should be no units in the body of the table. We should know that from previous study anyways. And the column headings should be fully descriptive and relate to the independent variable and dependent variable. So for example, a column heading wouldn't be time, it would be time taken for the solution to turn colourless in seconds. And the independent variable column heading wouldn't be concentration, it would be concentration of glucose solution or something along those lines, so that it's really descriptive, and from the table we can tell what the independent and dependent variables are. Tables shouldn't be split across pages because you'll lose communication marks for this, because that's incorrect formatting. Any anomalies should be identified. Now you can highlight them or circle them or identify them in any relatively sensible way you wish. And uncertainties should be in column headings. So where it says time taken for the solution to go colourless, in brackets you would then have seconds, and then you would have, for example, plus minus 5 seconds if that's your uncertainty. The process data, so in your raw data table, you literally just have the data that you collected. You don't have means, you don't have standard deviations, nothing, just your raw collected data. In the process data table, this is where you can put your means, standard deviation, and any additional calculations if you've got things like rate. Your process data table should also have units in the column headings and uncertainties if need be. Again, column headings should be descriptive. And then you should also provide sample calculations. So for example, if you have calculated a mean, underneath your process data table, you need a sample mean calculation, you need a sample standard deviation calculation. For standard deviation, you can use Excel if you want to, so you can reference that, but obviously make sure that it's referenced. And then your qualitative data should come next. It should be your written observations of what you've kind of seen and anything that you've recorded that isn't quantitative. So colours, smells, any aesthetic changes. So if something's gone cloudy, things like that. You don't need as many observations as you do pieces of quantitative data. So if you want to, you can group your observations into trends, or if you want, you can have a little observation for every single quantitative piece of data that you take, but be aware that you don't want the qualitative data to be taken up pages and pages and pages. So just be careful with the qualitative data that you provide. Okay, so this is an example of a table from a previous IA. So what's wrong with this table? We've got no descriptive column headings. We've no idea really what this is showing. You've got raw data and process data in the same table. You've got units in the body of the table. And there's only 16 pieces of data collected, so you need to have at least 25 pieces of data, so it should be 5 manipulations of the independent variable and 5 repeats at the very, very least. There's also no uncertainties outlined here. So let's have a look at what this table should have looked like. So here we have your solution. This should actually be more descriptive. You've got absorbance of solution using colorimeter with a wavelength of 680 nanometers. That's much more descriptive than first test, second test, third test, fourth test. And then you've got your 1, 2, 3, 4 repeats here. You've also got an uncertainty. Now, please be aware that in the body of the table, so because this uncertainty is 0.005, so you've got 3 decimal places, they need to be here as well. So you need to put a 0 on the end of everything so that it's consistent with the uncertainty. You've also got your uncertainty calculation here. So the smallest increment of the colorimeter was an absorbance of 0.01. What we then do is we split that. So 0.01 divided by 2 would give us plus or minus 0.005. That means that essentially if you collect a piece of data that's 0.720, actually what you're saying is this is the result that I got. However, I am aware that it could actually be anywhere between 0.715 and 0.725. So that's the uncertainty. We're kind of giving a bit of leeway. That's what we mean by uncertainty. So make sure that you include that, because that's mentioned in the marking criteria. Okay, the next thing is stats test. Didn't put a title here just to confuse you. So what I've done is I've outlined some of the common stats tests that you might want to use. Now, you are allowed to calculate stats tests using online calculators. Please steer clear of putting all of your data into an online calculator and then just taking a screenshot and not understanding what it's actually telling you. So it's really obvious when we mark IAs whether you actually understand the point of using the stats test and what it's actually telling you. So let's just go through one test at a time. So the chi-squared test is used to compare observed and expected data. So you use it when there's clear expected values. So for example, in genetic crosses, there's expected Mendelian ratios, or if you've got something with standard expected values. So for example, if you were looking at the frequency of different coloured Skittles in a packet, the manufacturer releases data telling us the percentage breakdown of each colour of Skittles in a packet, or what you should expect. So if there's expected values that way, then you can use chi-squared. Now what you then do is you put your chi-squared into a chi-squared calculator. You will get a chi-squared value. If your chi-squared value is higher than the critical value, you are going to reject your null hypothesis. So be aware that for the stats test, you need to outline a null and an alternative hypothesis before you do it. And then you need to state whether you are going to accept or reject the null hypothesis. The chi-squared value is going to be compared to a critical value. Now, the critical value, if you just Google chi-squared critical value table, you'll see that there is a standard critical value table. Now, you have to find the correct critical value depending on your degrees of freedom and the probability. You will always use a probability of 0.05. I shouldn't say percent there, sorry. A probability of 0.05 or 5%, because that means that there's a 5% probability the result is due to chance. For the t-test, this compares means of different sets of data. So if you have, in fact, let's go through paired and unpaired, and this might make it more clear. So in a paired t-test, it's where the same group undergo testing in two different conditions. In an unpaired t-test, it's where two unrelated groups are compared. So the same people aren't taking the same test. It's different people in different groups. Obviously, you don't necessarily have to use people or participants. It could be other discrete categories. And when you calculate your t-value, again, you have to compare it to a critical value. The t-test critical value table is different to the chi-squared critical value table. So if your t-value is lower than the critical value, the null hypothesis is rejected. And again, p equals 0.05, so there's a 5% probability the result is due to chance. But the third and final stats test that I'm going to go through, so you don't necessarily need to use one of these three. You can use a different stats test. I'm just going through popular ones. So Spearman's rank gives a quantitative value of correlation. So if you're looking for a correlation in your IA, then Spearman's rank might be the stats test for you. You use it when you have two ranked variables. So obviously you must be able to rank the independent and dependent variable. Your Spearman's rank value should be between -1 and +1. If it's not, then you've done it wrong. +1 means that there's a perfect positive correlation, whilst -1 means there's a perfect negative correlation. And if you get a Spearman's rank value of 0, it means that there's no correlation. So obviously the closer you get to +1, the stronger the positive correlation, and the closer you get to -1, the stronger the negative correlation. And obviously the closer to 0, then the weaker the correlation. I did put a title there. Okay, so Graphs. So usually 1 or 2 graphs is sufficient. Sometimes you get IAs that have like 15 graphs. I don't want 15 graphs. Just 1 or 2 graphs. Make sure that you choose the correct graph type and make sure that your graph is only process data. Do not plot raw data on a graph. That's of no use to anyone. We're looking for trends in order to draw conclusions in this IA. So you need to make sure that it's process data on your graph or graphs. Graph should be around half a page in size. I know it's easy to think, oh, I'll just make my graph smaller so that I save pages. Please make sure that we can actually see the graph. Make sure it's got a descriptive title. That should be easy. Label your axes with units. You need to add a trend line if that's appropriate to your graph type. If you've got more than 1 line or more than 1 bar on a bar graph, then you need to include a key. And make sure that you use Excel or another program to produce a graph. Don't hand draw it because it's 2020 now, right? And ensure that you have the correct axis scales. So here, you've got an incorrect scale on this graph. So obviously, if you change the scale, then the graph would be much clearer. And this is just a waste up here. These points are joined rather than having a trend line, so don't join the points. Make sure that you have a trend line. Identify any anomalies on a graph if you need to. There's no axes labels here. We don't know what that graph is showing us. And then there's no key to identify the different coloured bars on this graph here. Finally, this is the marking criteria for the analysis section of the IA. I'm not going to go through this, just pause it and have a read through if you need to.